Good afternoon to each and everyone present here. On the behalf of Scientific Committee, I welcome you all for this scientific session. Sir, on the behalf of Scientific Committee, we welcome you and sorry for the delay for some technical reason. Sir, we are feeling very privileged to introduce you. So, the speakers of today's scientific session is Professor S.S Sharma, sir, and presently he is working as a professor in the Department of Pharmacology and Toxicology at Mohali, Punjab, India since 2009. Before joining Mohali, he worked as a postdoctoral fellow at the USA and he did his PhD from the All India Institute of Medical Sciences, New Delhi. Sir has done his B.Pharm and M.Pharm from the Dept, New Delhi. Sir is a fellow of Indian Pharmacological Society. His research interests include understanding the role of pharmacological agents in a stroke, cardiovascular disease, and diabetic complications. Sir has published more than 150 research papers, review papers, patents, book chapters, and books in a national and international journal. Sir has more than 20 years of teaching and research experience, and he has guided more than 100 masters and postdoctoral and doctoral students. Sir has delivered more than 100 invited talks at national and international platforms. Sir has completed more than 20 extramural and industrial funded projects. He is recipient of various prestigious scientific organizations. He was the editor of Science Research and Information in Pharmaceutical Science. For now, he is on the editorial board of several international journals. He was the secretary of the International Pharmacological Society for 2008 to 2011. Sir has organized several international conferences, including Drug Discovery and New Millennium Drug Discovery Conference in 2000. Sir has organized G3NM2 in 2005. Sir has organized National and International Conference C3NM3 in 2000. Sir has also organized the 40th Annual Conference on Pharmacological Society in 2000. Sir has working as a co-organizing secretary in ISFCON 2016, the Stop India 2017, and the Stop India 2020. So we are very privileged to welcome you as a speaker for the scientific session. Today's chairperson is Dr. Puneet Bansal, sir, and sir, we are very happy to introduce you as a chairperson for the scientific session. Dr. Puneet Bansal is an associate professor at Head, Department of Pharmacology, School of Basic and Applied Science, Central University of Punjab. He has more than 12 years of teaching and research experience. Sir has completed his M.Pharm and PhD from the Institute of Pharmaceutical Sciences, Punjab University. His research the pharmacological screening of semi-synthetic and synthetic drugs in the animal model of various diseases like movement disorders, traumatic brain injury, epilepsy, patient cognitive impairment, and related disorders. Sir has completed two major projects, AICT RPS, first 20 lakhs, and PhD SPRP in Sciences Award, first 24 lakhs. One project is ongoing from SPRP PhD, first 32 lakhs. Sir has published more than 110 research papers in the prestigious national and international journals. Sir has also published 6 books and 22 book chapters. Sir has invited for various national and international conferences. Sir has guided more than 50 M.Pharm students, and currently he is guiding PhD scholars, 5 PhD research scholars. So we are very privileged to invite you as a chairperson for the scientific. And now I request Dr. Charan sir and Dr. Puneet Bansal sir to continue Dr. Charan. I would like to introduce Dr. Bharti now. Dr. Director, Scientific Advisor.
Afternoon, everyone. I think this particular conference on Impro, and this is brainchild of Dr. Charan Singh. I can see that actually when he contacted me regarding this particular meeting, I was very excited. He asked me, and he like, can you speak in this particular meeting? So I thought like it would be privileged because he was a own student in NIPA, though I am not writing him actually. But like since most of the students attended our classes, so that we know very well. And Dr. Anthi was like she was alumni of Department of Pharmacology. And I really appreciate, I think, I can think actually, I hope actually you are with your heart and brain sitting here.
Yes,
I hope it's not there in the culture program. That is good. It's still like, though the culture program is going on, until that tradition in most of the international conferences, actually, but the scientific sessions are given more priority than other things. But still, I think since you are here, I really appreciate your interest in the subject of Hebrew in this particular workshop. Uh, and like I can appreciate one of my pharmacologist colleague, actually, Dr. Bansal, and we are associated with him also in many ways. And being a neuropharmacologist, and I can tell you, like, I think today you have heard from my because like I did my doctorate with him at the team and then my neuropharm for more than 2 decades. I think, like, and the subject I thought I feel like let's talk in front of you and being a student, maybe you are in PhD or master's student, you can have take home messages so that in some of the areas you can very well work. I think like as a student of pharmacy, everyone knows about what is drug discovery, how it is being done. No need to explain to all of you because you all already learned B.Com and M.Com students that you know how the drug discovery is done and there are various various like process or maybe basis of the drug discovery. Do you know always that preclinical studies need to be done actually? Okay, and for preclinical studies, you require a expert chemist actually who can synthesize molecules actually, and you also require some people actually who are very good in pharmacoinformatics actually, drug design and all. So they're like I think medical chemists play very very important role in drug discovery. In addition, nowadays you can know that the people from natural drugs and people from like pharmacology, that area also play very important role because you can not only have the new molecule, you can also have some of the like plant extract for investigation, and in addition, like you can also have newer proteins like protein therapy, even this biotechnology drive product. Alright. So that's our lab actually work in all the areas. We also have a collaboration with the biotechnology department They are making some of protein therapeutics actually, and those are being investigated for their pharmacological activity, particularly in the CNS disorders. So you can have, as a pharmacologist, you can have a collaboration with Charan Singh also, because he can make new formulations for you. So he can make some newer formulation actually which can cross blood barrier, because that is one of the major challenge for drug discovery and development. Things like target selection, lead discovery, medical chemistry like synthesizing molecules, in vitro assays, in vivo assays, and then clinical trials. So most of the time, if you look at the drug discovery cost, 30% of the drug discovery cost, when we call about like $1 billion, about $300 million goes to your preclinical studies, okay, and then clinical trials, phase 1, phase 2, phase 3, 70% of the cost. $700 million goes into that one. That's because like doing research on the patients, there's a lot of GCP involvement, good clinical practice involvement, recruiting patients to the clinical trial, not that easy. That is why like 70%. Drug discovery is a major need of this century, because like if you see that in COVID, we have seen like many people who have gone into like got this infection, they would have gone into like some kind of psychological stress, depression, stress, schizophrenia. We see many of our students actually when they come actually and we feel like they are very depressed. But because I think like maybe sitting at home or maybe always with social media. Okay, that could be one of the reasons that everybody is just occupied, not interacting with the friends physically. Okay, and a lot of time goes into that. Stress is one of the and plus challenging always like stress, because like competition. If you are in one class like 30 students and you are competing, one only can be the topper. That's also a stress, but that's a very good stress if you try to be the topper of that particular class or that particular course. And if you look at the global CNS market, it is more than $80 billion in reaching, I think, by 2025 is going to cross more than $200 billion. So that's kind of like a lot of potential that they will have regarding these points. Based on the scale of the blockbuster drug and the current CNS drug discovery program, unable to meet the burden of CNS. So now the issue is that I think this topic is good, but how many people are working? How many industries are working in this area? If you look into this team factory, very good pharmaceutical was working in that area. Landmark was working, but again, stopped working. One of the company like predominantly like Suvan Life Sciences, they work in the area of the CNS. Very, very few companies. Maybe if you look at like academic institutions, most of them are working. But again, translational videos, translational research. Always you have to think of doing translational research, which can be from the bedside to from the bedside to the bedside. That is always, I think you just try to think from the market. Otherwise, happy actually to see that some of the students will be to formulation and marketing. So I think that kind of motivation you must take from that. Like when one of your colleague can do, I think you can also develop some CNS therapeutics, whether from natural products actually, whether from protein therapeutics, or whether from the new chemical entities synthesized by a very good chemist. That can also be. If you look at the CNS drug market, like you can see over here, like most of the costs actually involved in this particular line. Because one is the mental health, and if you look at the drug market, actually most of the time this fluoxetine or like those SSRIs used to be the top in that particular list actually. Because of like mental disorders are the stage one, followed by neurodegenerative disorder, CNS cancer, and then CNS trauma and the neurovascular diseases. Now you see, look at it actually. Sometimes like when I used to teach my students, I used to just put a lot of drugs related with the depression, schizophrenia, and all. But now you see, because of like I think when you look at the cost of the protein therapeutics, which is very very high, and that is why the drugs which are acting through that pathways are going to be on the top. You see that on the multiple sclerosis, this will be prophylactic. I'll tell you like one example, like where there is a need, like you would have seen the schizophrenic patients actually, and they require continuous treatment, and they cannot able to take the drug and all, like if every day, like 3 times a day, if they have to take, they'll forget of taking, or they will not. The serious condition is like that, they will not take the medication. So can you develop a formulation, like it's a challenge for even our friend Charan actually develop a formulation, like you just put the implant and for 3 months forget everyday administration. The same way for Alzheimer's patient, they forget to take the medicine. Develop a formulation of ISFCP or any of those which can be just put it once for 1 month, you forget, and all the symptoms are under control. So those drugs are not just symptomatic. Okay, they do not take care of the progression of the disease. That is also one of the major challenges that discovery, that progression of the CNS disorders, if we can stop it with that. Another challenge actually for pharmacologists and other scientists. These are the challenges in drug discovery, like complexity of the brain, billions of neurons, billions of like synaptic connections. They say really big challenge, and the signaling is very fast, actually more than what Shatabdi done on the display. Those kind of fast signaling, so much of network make brain is one of the highly complex organs, and it's require like 25% of your body oxygen and body blood glucose consumption. So even we are sleeping, actually our brain is working, at least consuming a lot of your oxygen as well as your glucose. So that way, I think we have to always think like complexity of the brain is one of the challenges. Blood-brain barrier is one of the major challenges. But if you stop everything, okay, stop everything means only very few things, like if it's a lipid soluble or maybe there are some transport systems are there. Anyone remember like how this whether this dopamine can cross blood-brain barrier? Anyone of you? No, dopamine, very good. Like dopamine cannot cross blood-brain barrier, okay? But it can cross blood-brain barrier when you're making it as a product. And what kind of transporter is used? Aromatic amino acid transporters. So this use some transporters to cross actually liver open can cross blood-brain barrier. So we have like several like transporters in our blood-brain barrier actually can actually cross some of the drug to brain. But again, actually, like you would have seen that many of the like very good neurotropic factor like BDNF, brain derived, whether it can cross blood-brain barrier. No, all the big molecules, protein, they will not cross blood-brain barrier. That's one of the challenge actually. But again, as a I think pharmaceutics person, you can have a kind of a Trojan horse technology. You try to add this kind of a bigger molecule with some kind of a domain actually which can be identified by these transporters. Okay, so that it can cross blood-brain barrier. So those kind of technologies are being used by many of the companies actually, and I think you can also use them in that. So that's like if you can make like superoxide dismutase cannot cross blood-brain barrier. It's very good antioxidant. You have to use, or otherwise actually another alternative is there. You can use SOD mimetics. SOD is a big enzyme, superoxide dismutase, very good antioxidant, will not cross. But if you make SOD mimetics, which are a small molecule but behave like the SOD, okay, can take care of those free radical generation like superoxide can be tackled by SOD. So that will like develop some SOD mimetics, develop some catalase mimetics. One of the molecule maybe you have heard of is Tempol. Tempol is having these kind of activity, like it is one of the like SOD catalase mimetic and can cross blood-brain barrier and will have the antioxidant effect in the brain that is required. Then this efflux transporter we mentioned to you, and patient heterogeneity. One of the major issue always that with the CNS disorders, like one patient is like responding to that drug and another patient will not respond to it. initially when Tacrin came for treatment of Alzheimer's, maybe now it has been withdrawn, but they have seen that like only like 30 to 40% patients were responding, other patients were not responding. So now like pharmacology plays very important role for understanding pharmacogenetic variation. Okay. So those variation, why like this particular patient is responding, this particular is not actually, and nowadays even like this SNP test, single nucleotide polymorphism, you can find out, and those are also coming up. And another area which has come up is regarding the precision medicine. That's also one of the like coming up area in India. Maybe uh, other countries were there for quite a bit, like for more than a decade. But now, like for many of even students, they starting developing companies like startups for doing precision medicine. So nowadays, like government of India also gives a lot of funds. Actually, you can have your own startup company, okay, with some good ideas, and you also can get some of the investors. Then big challenge is always like multiple targets because if you see the one disease condition cannot be treated like Parkinson. The levodopa is not helping. After 3-4 years, 3-4 years, like continuous administration of levodopa itself induces motor impairment. But dopamine, when it goes to the brain, converts into effective molecule, it also release some radical which can be damaging to the dopamine neuron. And that is why like dopaminergic receptor agonists have come up into the picture. You give levodopa with your dopamine agonist after 3-4 years. Or maybe add some MAO inhibitor like Saligate or multiple targets. That's also already there. So that is like as a chemist, you can develop some biofunctional molecule, the single molecule having multiple activities. It can have like your MAO inhibitory activity. It can have your like dopaminergic agonistic activity. Like that, you can also have a biofunctional molecule. Then CNS disease model is not always like one model will not work always. You always have to use multiple disease models, multiple models. One like Parkinson disease, MPTP induced Parkinson, not the solution. You need to do 6-hydroxydopamine. You can also do like free-form fibril, like this cyanogen fibril, and you can also do like transgenic animals. Okay? There's multiple models need to be used, whether in vitro or in vivo conditions. And what is the predictive validity of experimental data? And that is why you need to repeat it to the multiple points. Establishing biomarker of disease existence or disease. This is a very good area, and I think pharmaceutical analysis people they can play very important role. Okay, pharmacology students can give you the sample of like serum or CSF. Okay, in control animal and the disease animal. Then these analysis people like they can find out like biomarker. Actually, comparing these two things, they can go for 2D and electrophoresis, and they try multi-top down. They can identify different protein as a biomarker or so like that. So that's also a very good area for identifying biomarkers actually for any disease. And always in challenge when abuse liability and other side effects. Any CNS drug will going to have abuse liability. That is why all the drugs need to be checked whether they are having abuse. Otherwise, like this regulatory agency will not approve. You have to perform. You have to show like these molecules do not have abuse. So these are like some of the disease conditions which you see that the stroke, neurotrauma, epilepsy, Alzheimer's, Parkinson's, if you look at the prevalence, I think the stroke was like some time back considered to be the second leading cause of the death for the one. Okay. And the first major cause of disability was person who gets stroke generally goes into half of the body gets paralyzed. And that's very, very important area, I think, that we are going to discuss. And if you look at the number of deaths of the stroke, 6.55 million deaths globally and about 0.69 million deaths. Okay. Like about like 6.9 lakh people like every year die because of the stroke. Okay. And always a therapeutic window within 4 or 5 hours, 4 hours. The drugs which are available can act within 4 to 5 hours. After that, no treatment at all. That is the challenge actually for any chemist, any companies to develop this kind of funds. So that's like stroke status and economics is one of the major issue actually that we have to take into the consideration, and when it is one of the leading cause of death. So what you see that actually like I think stroke basically like most of the cases of stroke are ischemic stroke, wherever the blood flow of the in the brain actually like are generally interrupted. So what happen like sometimes actually there's a heart actually like if you're doing exercise, our heart rate start increasing. It will increase to 170 or so or 180. And always you have to be cautious also like always just put it from the 220 minus your age. So your heart rate should not exceed that one. Always take care. If I am 50 years actually like means 170. If I'm doing exercise on 170 plus, anytime my heart can get hit. Okay. So always you have to keep a watch on your heart rate. So during actually like these kind of active fibrillation or trigger, some of the thrombus which are formed in the body actually they will travel through your carotid artery. Like you can see like your one of the highly pulsatile artery, two arteries like right and left common carotid artery. These arteries actually supply blood to the brain. So there is internal carotid artery supply blood to the brain area internal and external carotid artery supply blood to the facial area. Okay. So this internal carotid artery and then there are two vertebral arteries. They supply and they form a circle of belief actually so that actually this solid blood circulation can happen over there. So in this case actually like if you just look at ischemic stroke, this clot can go actually and thrombus can block over here, then the blood supply to the brain. Or maybe another type of stroke is always a hemorrhagic stroke. The person is under tension actually, high tension, lot of I think everybody tension, but person is hypertensive, having diabetes and other actually, and the blood pressure shows more than 200 millimeter of mercury, the chances of some of the blood vessels can burst in the brain. And nearby like hemorrhage would be there, and 10 to 15% of the cases of hemorrhagic stroke are there, always there. So most of the cases like what we come across, this one is ischemic stroke and other 10 to 15%. But what you need, it is an emergency and you require immediate treatment. So very well, I think always you remember this FAST fast. Okay. I think why you have to remember because you can help your nearby or anybody actually just passing on the road actually somebody facing that symptoms that you can actually oh this is a person is having a stroke and it's an emergency and you inform nearby hospital location emergency. What it indicates like this smiling is one side grouping. Okay. Like you can see this smile is one side grouping. If you ask to like raise the arm, one will like just go down. Okay. Or cannot stand like that for normal person. Go down. And then slurred speech. The way like anyone is speaking like very clear speech, you will think like that person is drunk actually. Okay, that kind of speech actually they will have. But if they have actually like slurred speech, they are not able to speak clearly. You are not able to understand. And T is the time to act. Very fast actually. If you can take the person to the hospital within 4 hours, they can be administered thrombolytics. Okay, they have a life out. These thrombolytics have half-life or maybe therapeutic window of around like 4.5 hours. Various diagnostic tools are being used like brain MRIs are being done, echocardiograms are being done at like okay, then all those blood pressure monitoring needs to be done at like and their blood samples are taken. As you can see there, this is kind of a carotid Doppler. Most of the time, like after 40 years, everybody say, okay, go to the doctor, try to get it checked up. And before getting a full blown stroke, some people also get TIA, transient ischemic attack. They will get actually attack for brief period, like blood vision for 1 minute or half a minute. Okay, and some kind of dizziness actually. Somebody feeling actually these kind of symptoms, they can be warning signs for the stroke. That also needs to be taken. very well like normal agitations and neuroactivations actually. What we have actually, as I mentioned, like to go quite low. They can be given as a clade, otherwise like neuroprotective. That image there is a need of research actually can be developed neuroprotective agents which can be given after 20 hours, 24 hours, after 48 hours. You have seen our rural areas. Patients cannot travel to the hospital. Maybe in the night, everybody wait. Okay, tomorrow when daylight will be there, then only will go. But time is lost because neuronal damage have already started and can lead to a lot of damage. This kind of like you see that ischemic core and the area. This is the core actually, which is already impacted area. You cannot reverse. Okay, no drug to reverse impacted core. But in case of like over here, this area, what you see, can be reversed if the treatment is started on time. And this area is called penumbra and can be very well reversed if you give some neuroprotective agent. Japan has already approved one agent like Idarawan. Okay, this particular agent can be effective, but like other countries still have not approved for this for the stroke treatment. Now you can see it's kind of a blood flow. Actually, here's the blood flow not there, but after giving this thrombolytic, the blood flow there. So there is always need of neuroprotective agents and always like you have to target like which pathways of brain damage we can work on. So if you see that the events actually how they happen, now you see that the moment the stroke is there, okay, there is excitotoxicity of the brain actually. And this brain excitotoxicity is going to lead for several days. And then you can see that the inflammatory changes and as well as the complications which are associated with that. So these kind of like some of the events actually I think most of you would be remembering that I think very common to each one of you that these can be targeted in case of a stroke. And what you can see the excitotoxicity because whenever there is blockage of the artery, immediately the glutaminergic neuron actually there is a release of like such of glutamate. And this glutamate acts on the glutamate receptor and can lead to the neuronal death. What you can see also there is inflammation, calcium blood, oxidative stress, neuroinflammation, regeneration, and apoptosis. These kind of pathways have been implicated, and many of the scientists are trying that. Even like we also have a stroke research facility, and we created, and we are doing stroke research. I think it was being done initially with Professor Vaidya Gupta lab. Then we also have set up very good facility for stroke research, and that is being done. So now we see kind of a model actually. As a student, I think I just prepared this lecture for the student, like how they can develop the stroke model in animals. You see that like what we are looking at that is blood supply to the brain, and one of the major arteries which supplies blood to this cortical region is middle cerebral artery, MCA. So what you need to do, like I think you need to block this middle cerebral artery so that you can develop in animals. And what we have done actually, I think we have this kind of a isolated carotid arteries, okay, and then internal carotid artery, and then it has gone into I think this is a really great opportunity for me also to speak in front of Professor Vaidya Gupta because this is the model actually like was developed in actually and later on like we developed in Mohali. So it's a I think great tribute to you, sir. So this particular like middle cerebral artery actually like that we are blocking and that induces the damage to the brain. And what we have used actually we use like a stereotactic apparatus. So even we can record cerebral blood flow using laser Doppler blood flow monitoring system. And you can see over here, like when we have initially it was 100% blood flow. Now we have blocked it at this particular time. Now you can see the blood flow have come down. So now like around 80% blockade is there, like because the blood flow from 100% to come came down to 20 means 80% block. And then for 2 hours this block was there, and then it was actually repurposed. And along with that, actually, we have measured all the parameters like using the blood gas analyzer and physiological parameter monitoring system. And I have seen actually even ISF project on Moga also have those power lab system for measurement of physiological parameters and all. That's really wonderful. And these kind of like information studies are being done using TTC staining. And what you can see over here, like this is the TTC stain sample of the sham operated animal. If you're a pharmacologist, you know what is the sham operated animal, like in which you do the surgery, but you do not block the artery. And in another case, like this is one in which blood supply was blocked and using that MC occlusion, and this white portion is kind of a blockade of the blood supply. And then we measure all like neurological function tests. And what we have initially investigated, like some of the pharmacological agents, and we targeted to this peroxynitrite pathway, poly-ADP ribose polymerase pathways, using different interventions which are available. Peroxynitrite expressors like FETMPYP and FETPPS, two of these molecules were investigated. In addition, like this path inhibition, poly-ADP ribose polymerase inhibitor, like NU1025, nicotinamide, 3-aminobenamide, 1,5-isoprolyl and 4-amino-L-naphthalene. So those kind of agents initially investigated, but if you are a chemist actually and you really would like to work in the area of stroke, you can make the derivative of these agents, and then those can be investigated by a pharmacologist sitting in this particular room. And these kind of like the therapeutic window studies we have done at different time points. These agents were investigated like 2-hour administration, 6-hour, 9-hour, because always like we would like to see the therapeutic window, because the therapeutic window for thrombolytic agents is quite less, around 4.5 hours, and always we need an agent actually which has a better therapeutic window. So these agents actually we have seen that in function was very high in case of like the group where the MCO was blocked, and you can see this treatment has reduced the function. So this indicating like this particular FETMPYP and FETPPS, they are having wonderful effect on in function. And then we looked into like this peroxynitrite expander, whether like they are inhibiting generation of peroxynitrite tables actually. So this was done using glutamine 1,2,3-fluorouracil, and you can see that in the sham operated compared to your cultivated, where the ischemia was induced, there was increase in the peroxynitrite, and that was reduced by this FETMPYP and FET. And then you can see over there like neurological score was reduced actually by these agents. These results are indicating that like there is a neuroprotection which is there, but they have also reduced the neuronal cell viability was improved by this. Ischemia generally like reduces neuronal density, and it has been improved by, now you can see it has been improved by FETMPYP and FETPP. And we also looked into like DNA fragmentation, because I see most of the paper, even from Moga, I get a lot of paper for review, and we find they are doing even DNA fragmentation kind of studies. And what you can see that kind of a FITC fluorescence, where like you can see ischemic people within animals, you can see a lot of fluorescence, but it was reduced by FETPPS and FETPPS different doses like 1 mg and the 2 or 3 mg per kg dose, and those kind of classes. And then we have done like multiple of these poly-ADP ribose polymerase inhibitors, and you will see that various agents like nicotinamide, 3-aminobenamide, isoprolyl, 4-anide, they showed very good effect over there. And NU1025 very selective actually. Even some of the clinical studies actually are already being done in some of those. The edema and neurological score was improved, and we also looked always the repurposing. Actually, can the existing molecule can be repurposed for other disease conditions like PPR gamma agonist, like pioglitazone is already available. Actually, for like whether it's possible over there or not. So now this pioglitazone, along with that, we also looked into GW1929. This is a compound from Welcome, and this particular compound has one advantage in non-acidulin ion. The side effects which are associated with the pioglitazone would not be there with the GW1929. And we can see that like 1, 3, and the 10 mg per kg dose has significant reduction in the neurological damage associated with and the blood flow, effect of the blood flow, you can see like there's no change in the blood flow, means like it's not basically improving the blood flow like in case of thrombolytics, they do a lot of blood flow. PPR gamma agonist on inflammation was done, and you can see some of the inflammation markers. That is why I was telling like some of the biomarkers you can develop, and TNF alpha, interleukins, and all these are the commonly used biomarkers, and many of you are also doing in focal as well as imagery. And you can see over here like kind of a DNA fragmentation which has been taken care by biological. Apoptotic DNA fragmentation, actually you see the number of like cells which have been actually like there at the cortical and the subcortical neurons. Actually, those kind of dead cells can be improved by this particular. Another model actually like what we use is global cerebral ischemia model. The commonly used model is in Dept. The advantage of Dept is that like because in human, in other animals, you have to block at the 2 carotid artery and 2 vertebral artery. Reaching vertebral artery and doing surgery on vertebral artery is always very, very difficult. Lot of mortality. In this particular model, just blocking common carotid artery can induce global cerebral ischemia. This is the model actually the Dept, which is being used actually where we are blocking like common carotid artery, and that can induce the blockage of area and leading to global. These kind of like experimental design are being followed actually. 90 up to 96 hours we do all the neurological function tests actually. Neurological scores are done at different time points, and the treatment like we apply all kind of paradigm like treatment as opposed to. So this particular molecule which I was mentioning like GW1929, we also investigated like as I mentioned that any molecule actually just one model will not be sufficient. You need the multiple models. If you look into some of the STAIR's recommendations, actually they are indicating that you need to evaluate your molecule for neuroprotective activity in different models. We did in the focal cerebral ischemia model, then followed by the global cerebral ischemia model, and we also find very good effect in this particular model of hippocampal damage. Actually, you see hippocampal damage actually was like you can see in case of like GW19 at different time, different dose, and they are very good at it. And same way DNA fragmentation in checking hippocampus, actually you can see you can do this violate actually hippocampus, or you can do like kind of this kind of study and you can find. So this review actually like we publish in News and Perspective, and we will put all the mechanisms which are the PPR gamma and the so now I think this is the one actually like what I was telling because I see a lot of paper and research work is being done in ISS Moga. Chemists can synthesize new molecules in a hyper molecular chemists actually they have synthesized some of those molecules, and those molecules have been investigated for CNS because that is the advantage because you can get patent. Because I appreciate really like last year I attended and I took Viva of some of the pharmacology students. I have seen like many of them they have a publication as well as they have a patent. That's very good culture actually, and that should be continued actually, and that should be promoted. That's really wonderful. In our case, I feel like we have done multiple molecules were investigated to CNS activity. You can see the number of molecules were investigated to CNS activity. One of my colleagues, actually, Dr. Rahul Jain, in medical chemistry, synthesized some of the molecules which act for analytic activities, and you can see 35 of them are doing analytic activity, and 31 no analytic activity, and others were showing again actually stimulatory effects. And some of them we have taken for investigation into stroke model, like NP47, 355, 81, NP89, NP10 from micro is there. So those molecules actually like one. In addition, like we also do like safety pharmacology profile of these molecules, whether these molecules are safe for CNS. And as I mentioned in my the challenges, you also need to do the abuse value. You can go through the ICH website, ich.org, where you can find ICH S7A, S7B guidelines. Okay, and those guidelines will tell you like what kind of safety pharmacological studies you need to do for this one. And our laboratory is doing like core battery safety pharmacology, cardiovascular, CVS, respiratory, follow as well as the supplemental safety pharmacology. Studies are being done with this one. This was the center actually was given by like PRDP around like 1.78 crore actually we got for this one. So if you look at like so there are several, as I mentioned, the challenge, several agents were tried, but most of them were failed actually during phase one, phase two, phase three. It was the challenge. You can see nimodipine, NX5059, Trilajate, Bromethazole, MK01, Eptiganil, CGS 19755. So like a lot of molecules have been synthesized and they have been taken out actually. And they have been withdrawn with big loss to the company. And even you see like it's still like many of those molecules they are into the clinical trial. So several clinical trials are still going on, phase two, phase three clinical trials are being conducted actually. So then if you go to this website, clinicaltrials.gov, you will find a list of the agents which are in the clinical trial for this group, and you can find many of them. But again, like most of the industries, they don't want to work in this area because drawing competition. More than 100 molecules have reached clinical trials, but all of them fail. Very few, one or two or three, cerebrolysine, Vidaravon. Okay, only very few agents which are there in the market and are being used for the treatment. They're like these are the challenges uh, uh, from banks to bedside actually. Uh, why it's not happening? So always like our target is not very well validated. That could be one of the reasons. Biomarker, like we have the perfect biomarker for stroke or neurogenetic experimental models. The models which are being used really like is not single, multiple models. I just put two models in stroke, but there are other models. You can use that one, okay, to prove the benefit. Ethical challenges are there always. Patient is already half the body is paralyzed, and you are asking to try a new drug. Ethical challenge. And the clinical development is not that. I can mention like one of our collaborators, Professor Anil Gulati. Uh, I think I was introduced to Professor Gulati by our sir, Professor Vaidhi Gupta. In the Midwestern University, like he's there, I was doing postdoc at the time. I know him. He has developed a drug actually which has reached to the phase three clinical trial. And that compound was IRL 1620. That was a peptide. We also did a collaborative project, consultancy project in our lab actually, and now that molecule in the phase three clinical trial. So I think sometime if he's coming to NIPER, I'll try to bring him over here actually so that you can get a direct connection with him. But he is one of the pharmacologists who has discovered a drug, developed a drug, and in the market. So that kind of insight. So again, like future perspective, always to have like in this particular area, very good future is there, but you have to always focus. Translational approach. achieving these goals, actually you have to always look into the translational challenges, or you can meet them. Okay, you have to deepen your understanding of biological pathways in the area of stroke or brain repair. Clinical trials should be designed around principle of translational research and advancement in neuroimaging. So now I think like you would have seen like human MRI, but I think Professor Y.K. Gupta he did some of the MRI experiments with adenosine and all and showed zero protection with that one. So those MRI are also available actually, maybe costing around 7 to 10 crore rupees or so. Okay, but many of the institutions actually like IIT, Madras and many AIMS actually they are having animal MRI system and those can be. It's not only now the era is changing. I think everybody was talking about like just or biotherapeutics. Now you have to look into also medical devices. Even like Mohali last year, I think they started MTech program in medical devices. So can be developed like medical devices for CNS disorders. Okay. So it is not related to this particular lecture, but I still would like to just mention that this is 10 crores. So now like this particular like device can improve the quality of life of the patients with severe upper limb paralysis caused by the motor neuron. And you can see over there, like they put kind of like some of the electrodes in the brain actually, and that can monitor and they can help with the movement of it. So they maybe like Parkinson's disease again, like electrode can be implanted and trigger it with you. If you are getting a freezing cough wheel, you can just press that trigger and you will get affected. So maybe like future is there, and I hope Dr. G.D. Gupta can also start a course on medical devices in future. And another like you see the neuroprosthesis. So now you can see that a particular person actually like can talk. You were not able to speak, okay, but now he can, he or she can understand very well. And how are you? Those kind of devices are being developed actually and going to be going to make a boom in the future. One more, even like the blind person, visual prosthesis for completely blind people. That also being developed actually like they can see the nearby. Those maybe you require some engineers and all actually for develop, but I think in future we are going to have. So first of all, like I would like to acknowledge my mentor, Professor Y.K. Gupta. I think and I really privileged like in front of him actually I'm giving this talk. My big thanks to and funding agencies like Ministry of Chemical Fertilizer, DSC, SCRB, CSR, by collaborators from different universities like from NIPER, IDEA, from NABI, then Anil Gulati from different university. Okay, and he has got a company, S.S International. So now he is superannuated and he has a company. And all my students, all my favorite students, because they are really the hard worker and whatever I have presented is because of them. Because what they have carried out. They have, I think, put all their sweat actually in doing all kind of research and just presenting. With this, I would like to thank all of you, and I invite you to NIPER Mohali. If you are visiting anytime to Chandigarh, you have a chance to visit. Thank you very much. Yeah, yeah, please. Question.
Actually, failure of the all media
Yeah,
the
yeah.
probably the crossing of the drug across the region and I basically got down.
Oh,
So
yeah,
I
sure.
think in this manner, everything is going very excellent. We are doing the preclinical,
Okay.
but when we are going to the trials,
Yeah,
so the challenge, success rate is very
yeah, yeah. That is why, like, I think initially I mentioned all the challenges. What are the challenges? Well, like, what you mentioned, like, some of the challenges I mentioned, like blood-brain barrier permeability. So whether the drug was estimated, whether it was reaching at the site of action. Okay, that is very, very important actually. Most of the time, like drugs show very good neuroprotective potential, but it was never checked actually whether it's reaching at the site of action or not. That should be done. Actually. That's one of the things. And also like this model, unpredictability of the model. Like kind of a clinical situation, like always you cannot actually generate the thing or induce the similar kind of situation. Why? Because like in model actually we control everything. Okay. In patient when they come, they are having hypertension, they have diabetes and all actually. So always like I did not show my part actually. We have actually developed stroke model in diabetic animal. So we also develop a stroke in the hypertensive animal. So that also needs to be checked actually, like comorbid conditions. Model also needs to be in the comorbid. And another thing I think we should follow OECD principle of GLP whenever we are talking. And Dr. Y.P. Gupta is the father of GLP in India, and I think you have a great person. You can always like get to GLP there, and he's the chairman of GLP Technical Committee of PSD. I'm also a member actually, but again, anytime this GLP is required, you must inculcate. Each one of you should inculcate GLP in your practice. I think today go back and read GLP. This can give you really the work actually it can be reproducible. And that was a major challenge actually because initially like whatever the work was done in the animal actually that was not able to be. Okay. That is also need to be. Yeah, yeah. Now, yes. I think this is like valid question. I think depending on where the animals are available, everybody is taking. And even the genome analysis of these animals, like suppose every time when you can get some of the breeder from one very good source where like complete genetic analysis will be done. Vendors would be there. Always like people actually just get those strain actually and then they develop in their country. That would be always done. This like a lot of because you know that pharmacology, a lot of variations are there. Whether it's say like temperature, humidity, strain, some people use this stuff, some people use like spectrology, a lot of variation there. Pharmacological variation is under your control how you can do. Or maybe like if you're getting animals from other source, you try to compare the results of earlier one with that one, whether those animals are good. But that can definitely lead to variation in the result actually, and that even like robust standardization is required right in every case. Using a standard also in most of the cases because it's most of the people are not doing. But sometimes like animal ethics committee also every time do not approve the animal for standardization. This is also another issue actually which is there. Uh, so that is there actually, yeah. So that is why it's like I think what we do actually we also measure the cerebral blood flow. Like if you're blocking a both carotid artery actually not what you do already. Just check at the consistency issue always like the student, yeah, it's always depends on lab to lab because students keep changing. New students, you have to train every time new students. The PhD student, we generally give this kind of topic to PhD students who will stay with us for 5, 6 years and all. Okay, so that they will show the consistent results related to their implementation. And always like third party check also need to be done. It's not only what you have shown neuroprotective potential. Maybe I can send to other lab working in the area. You just check the neuroprotective potential of these molecules before taking them for any further preclinical or safety or other. That also is required. Yes. Yes. Yeah, there is no question from students. I think they should be more. Yeah, you're free to ask me or my yeah, I think by giving this lecture, I think the Ibro conference has started really because this is a brain research. When you call like international brain research organization and getting a grant for Ibro conference, I think it's really not that easy. It's a tough job. I appreciate, I think, the organizers for this. Require a lot of things to do. Yeah, it couldn't write. Otherwise, you can write to on my email ID. I think this S.S Sharma at the rate of nyco.dc.in. Okay, please be in touch with me. If you have any question and anytime, any help.
Thank you so much, sir. And it's always motivating and inspiring to listen you. And so far, myself is concerned, so I am hearing you since last 13 years. The story starts from 2009. I joined Mohali. So it's not difficult, but still I would say it's very difficult to compile the entire lecture in a couple of minutes. So students, so far the technical session of day one is concerned. Professor S.S Sharma explained beautifully all the CNS drug discovery and development related challenges and up to the final future perspective. So if you look at the presentation of the content of the same, then it starts from the challenges that where Sir has discussed about the blood-brain barrier, followed by various prevalence or the statistical data all over the world. Then the pathophysiology of the disease, followed by the detection and diagnostic test, and the treatment of the same disease. So sir, it is very nice to see the models which are being explored preclinically in the lab. So students those are working in the pharmacology, even in the pharmaceutics also, they are encouraged, and I myself encouraged to look into the models so far the stock is concerned. And secondly, sir, uh, whatever the content you have presented by taking some of the molecules into the experimental studies, it's again inspiring to look in. And so far, neurotherapeutics are concerned. So from the dice of this dear college, I try to convert the neurotherapeutics into nano neurotherapeutics, and that is, I think, the theme of the pro school also, wherein I proposed nano neurotherapeutics in the brain diseases. So conclusively, it's very interesting to again have a data or the content of the same. So finally, I request the dignitaries, Professor Rahul Deshmukh, Professor Puneet Bansal, Professor Michael Guptadi, and Professor Giri Guptadi to present the certificate of appreciation as a token of love. Sir, from your side, is there any comment? So please, you are welcome.
Good afternoon, dear students. Just I want to conclude whole lecture in this slide. This is not a slide. This is a 10 new projects for you to look at each topic. It will be a new project for next year from ISF College of Pharmacy. So I really thanks Professor Sharma sir for giving such an informative lecture. That always I feel is always knowledgeable for me. Not my first time that I listen to lecture, but it's always knowledgeable. And I ask to you that please choose your next project from all these topics, because we know everything about brain, but we don't know anything about brain. We know there is some therapies available, as sir said, but that's only for the symptomatical treatment. There is no cure for any CNS disorder. There is a really need of the art. Please work on it. Thank you, sir.
Dear students, I was very surprised that none of you had a question. I think this was a great opportunity that one of the great mentors came in, and still you are not utilizing its resources of knowledge. Is not big. Uh, I request all of you, please don't hesitate while putting up your questions, since these are the pillars of pharmacology, and they have done a lot of work in research. Whatever the doubts you have, you can put up straightforward there, and they are so humble. They will respond you very humbly and clear your doubts also. So the hesitations, I understand that you do have the hesitation, and but because of these people, so we also learn. I body was only. I saw Professor Gupta sitting over there. Still, my hands were sweating automatically. Sweat's coming. Since I just remember the day I faced my PhD synopsis presentation, I was not knowing. मेरे गाइड ने मुझे कहा मेरा मेरा कल मेरा चल तो हम गए हमें डर था कि कौन आ रहा है वहां तो हमारी यही दुआ थी कि प्रोफेसर गुप्ता सर और मेरा बैड डे वही था वहां पे कांच से देखा मैंने और वहां पे गुप्ता सर बैठे थे मुझे लगा कि मेरे लिए नहीं आए क्योंकि वहां पे दो और पेज के तरफ थे मुझे फिर भी लग रहा था कि मेरे लिए नहीं है तो but unfortunately the way haven't came and I have been pushed in front of you जो सुना था वो सच्चाई में बदल गया 2 घंटे तरफ चला हमारा पता ही नहीं चला शुरू कब हुआ खत्म हुआ ऐसा फीलिंग आई नहीं लास्ट फीलिंग नहीं मगर जो उन्होंने क्वेश्चंस दिए शायद उसी वजह से मेरी पीएचडी अच्छी तरह से हो गई क्योंकि वही बेस था वहां पे जो क्लैरिटी आई उनके क्वेश्चन से उसी की वजह से आज भी हमें क्लैरिटी है कॉन्सेप्ट्स की भले ही वो मेरे गाइड हो प्रोफेसर मनजीत सिंह प्रोफेसर शर्मा भले ही मेरे ऐसे बीइंग एकलव्य आई गॉट अ लर्निंग फ्रॉम हिम तो ऐसे टीचर्स की वजह से ही हमें ये बहुत अच्छी नॉलेज मिली तो आपको भी ये मौका है इब्रो ने आपको यहाँ पे एक अच्छा प्लेटफॉर्म दिया है प्लीज कम अप and ask your question. Thank you, sir, for a nice presentation. Thank you.
I think you must feel this particular thing there that you have missed something to ask from Sharma sir. Right. So I was there to have dignitaries on the dais for presenting as a certificate of appreciation in terms of a token of love. So I request the same to proceed further.
Without that, this group lecture cannot be completed. Organizing secretary should come and think it is you all right. I think organizing secretary should be given some privilege.
request the dignitaries to stay over there. So please, the next certificate or appreciation is from for Professor Desmond. So I request the dignitaries to do it. And for the next one is for Professor Benson. Thank you, sir. Sir, if the time permits and Professor J.D. Gupta, okay. So, from the core of my heart, I like to thank the participants as well as the organizing committee and the head of the institute, uh, dignitaries from the various institutions, the National Institute of National Importance, Institute of Eminence from various universities. So finally, I would again thank all the participants and the galaxy of scientists for happening this particular thing and material or to materialize the same. Today we conclude the day one of IPRO spotted meeting and workshop on geographies. So see you soon on the next day in terms of online lectures. So I again thank the organizing committee. Okay. So far, day two of the same meeting and workshop is concerned, then we have some speakers from Planning, then NBRC, National Research Center, then we have international speaker from University of South Wales, Australia, and the fourth one is from Central University of Rajasthan, and finally Dr. Aarti from the same college will have some scientific talks, and they will be speaking on the neurological disorders and the porous materials in the drug delivery and imaging. So I hope you will be joining the same through online mode. So this is all about the day two lectures. Thank you all. So all of you are invited for a high tea. Thank you.